Bismillahirrahmanirrahim. Welcome again to another new video. I'm going to talk in this video about vitamin D as a nutrigenomic. Nutritional genomics or nutrigenomics is the field of study that investigates the comprehensive effects of diet on our epigenome and transcriptome and their effects on our well-being and susceptibility to diseases. The word genome means all the DNA found within the nucleus and mitochondria of somatic cells. So it serves as a complete set of genetic information. Transcriptome is the primary outcome of this genome activity, which refers to the RNA molecules produced from those genes. The term epigenome originates from the Greek word epi, meaning above, the genome. The epigenome includes chemical substances that alter the genome, providing instructions on what action to take, where to do this action, and when. Different tissue cells have their unique epigenetic marks, contributing to their specialized functions and characteristics. The molecules obtained from our everyday diet constitute the primary environmental factor that we choose to take. Numerous macronutrients and micronutrients found in our diet, including lipids, lipophilic vitamins, serve not only as energy reserves but also have intracellular and intercellular signaling properties. These properties play an important role in regulating various physiological processes such as cellular metabolism and growth. And certain compounds among these substances can directly influence the expression of genes, whereas others require initial metabolism to modulate the activity of the transcription factors or chromatin modifying enzymes. One interesting example is the micronutrient vitamin D3, which can be obtained from specific diets like fatty fish, as well as it can be synthesized internally through our skin upon adequate exposure to ultraviolet B radiation. Significantly, when vitamin D3 is metabolized into dihydroxy 1,25 vitamin D3, it functions as a potent ligand for the transcription factor vitamin D receptor, so it exerts direct effects on the epigenome and transcriptome at a wide scale. Vitamin D directly affects the epigenome and the transcriptome of cells expressing the vitamin D receptor in various tissues and cell types. Through a ligand-dependent mechanism, vitamin D receptor binds to numerous genomic regions and via local modifications in chromatin affects the expression of hundreds of primary target genes. This demonstrates the direct effect of vitamin D on the epigenomic and the transcriptomic profiles of vitamin D receptor expressing cells. 1,25 dihydroxy vitamin D3 plays an important role in vital physiological processes such as maintaining calcium homeostasis for proper bone mineralization and influencing the immune system by promoting innate immunity and preventing excessive response from adaptive immunity. Also, vitamin D has been implicated in various other processes related to both health and disease. For example, it is suggested to potentially slow down cellular senescence or cellular aging by decreasing oxidative stress. Vitamin D deficiency significantly increases the risk for musculoskeletal disorders in children, rickets, and adults, osteomalacia, and fractures. Also, vitamin D insufficiency or in the worst scenario, deficiency contributes to a number of immunological disorders such as multiple sclerosis, rheumatoid arthritis, inflammatory bowel disease, type 1 diabetes, and also is associated with the severe complications from infections as intracellular bacterium, Mycobacterium tuberculosis, influenza virus, or severe acute respiratory syndrome, as we have seen in coronavirus type 2. There are many factors that lead to vitamin D deficiency or insufficiency, and from them is that living in northern hemisphere, especially in winter time, where the sunlight reaching the surface is too low for vitamin D3 synthesis. That's why the migration of humans from Africa, as well as modern lifestyle characterized by mainly indoor activities, lead to global vitamin D3 deficiency. And because the contents of food from vitamin D is very low, there is a strong need for vitamin D supplementation either in food or by pills. Among the approximately 1600 transcription factors encoded by our genome, vitamin D receptors has a distinct characteristic that make it special from this extensive family of regulatory proteins. Its activity can be directly modulated by a small lipophilic molecule, which is 1,25-dihydroxyvitamin D3, and this feature places vitamin D receptor in a similar category as receptors of estrogen receptors, ESR, or glucocorticoid receptors, GR, which have a significant medical effects due to their activation by female sex steroid estrogen or the stress hormone cortisol, respectively. Chromatin, which consists of genomic DNA and histones forming the three-dimensional complex, serves as the physical land for epigenetics. The position of the regulatory regions within chromatin, whether in loosely packed achromatin or densely packed heterochromatin, plays a critical role in determining gene transcription. So we can understand that chromatin accessibility emerges as the primary factor that affects gene expression, as it facilitates the binding of the transcription factor to enhancer regions and RNA polymerase to the transcription start site regions, also known as core promoters. Chromatin accessibility is regulated across the three major levels of the epigenome, namely DNA methylation, post-translational modification of histones, such as methylation and acetylation, and the three-dimensional structure of chromatin. And we have two types of chromatin by this classification: achromatin, which is predominantly located in the central region of the nucleus and is characterized by histone proteins that are largely acetylated and genomic DNA with low levels of methylation. On the opposite, we have heterochromatin that present an opposite profile, often sitting near the nuclear membrane and consisting of methylated histones and highly methylated DNA. In this diagram, we can see the epigenetic layers. We have three different layers of chromatin organizations, which is level 1 DNA methylation, level 2 histone modification. Histone is the protein where the DNA go around it, and the third level is the 3D chromatin organization. So these three layers represent either heterochromatin or achromatin. In achromatin, which is near to the center of the nucleus, and we have less methylated DNA, and this is an active one, which is more active in transcription, while heterochromatin contain densely methylated DNA and sitting beside the nuclear membrane, and this chromatin is inactive. Epigenomic programming primarily occurs during embryogenesis, playing an important role in determining the development of various tissues and organs. However, epigenomic programming also takes place during the differentiation of adult cells, such as in the continuous replacement of bone marrow, colon, and skin cells throughout life. It's important also to remember that the epigenetic changes do not involve alterations to the genome itself and are generally reversible in nature. Epigenetic changes frequently arise from signal transduction cascades initiated by external signals like growth factors, cytokines, and peptide hormones. Typically, a transient signal leads to a temporary epigenetic alteration. However, with repeated exposure, signals are more likely to induce persistent epigenetic changes. Consequently, patterns of histone modification or DNA methylation can stay for extended periods ranging from days to months or even years. As a result, the epigenome can retain the effects of cellular disturbances in the form of epigenetic drifts. In summary, the accessibility of a chromatin serves as a critical epigenetic factor in regulating gene expression. External and internal signals, including those originating from exposure to nutritional compounds, have the ability to initiate a comprehensive programming process across the epigenome. This process can result in a long-term memory formation through persistent alterations in chromatin structure. So micronutrients such as vitamin D3 often raise questions regarding whether individual serum levels are adequate to obtain the optimal health benefits. Also, there is another question about the inquiry about whether there are variation among individuals in their requirements for the micronutrients, and this will lead to the potential need for personalized supplementation. Human genetic variations manifest through approximately 4 to 5 million single nucleotide variants, 0.7 million insertions, deletions, and around 1,000 larger copy number variations. And among these variants, only a small fraction plays a role in the susceptibility to common diseases or affects the response to natural and synthetic signal molecule. Upon investigating the effects of vitamin D3 supplementation, it was discovered that individuals present personalized responses, resulting in categorization into high, mid, and low responders, as suggested by some researchers. This diagram illustrates the suggested classification of people according to the response to vitamin D. To get a personalized vitamin D supplementation in order to obtain a maximal clinical benefit of vitamin D, which include healthy bone, controlling bone homeostasis and calcium homeostasis, and muscle strength and development, and also the immune system through stimulating of innate immunity and preventing overactive adaptive immunity. And to get these benefits, they suggest that people can be classified into 3 groups. There are low responders, mid responders, and high responders to the same vitamin D level. So the concept of vitamin D response index involves assessing an individual's specific response to vitamin D3. By determining this index, personalized supplementation with vitamin D3 can be applied to achieve the optimal clinical benefits, including the prevention of conditions such as osteoporosis, sarcopenia, and autoimmune diseases. The molecular basis of vitamin D response index is not yet fully understood. In comparison to the finding about warfarin, it may be primarily explained by genetic variants. However, in fact, variations in the genes involved in vitamin D transport and metabolism can explain some of the inter-individual difference in vitamin D status. Like for example, the conversion of 70 hydrocholesterol to vitamin D3 on the skin depends mainly on a certain gene. So some people can form enough vitamin D even with low exposure to the sun, while others can't. However, the vitamin D response index appears not to depend on 25 hydroxy vitamin D3 serum levels of the investigated individuals. It means that it doesn't depend on respective genetic variants. To summarize, the field of nutrigenomics related to vitamin D responsiveness indicates the potential for personalized recommendations for vitamin D3 supplementation. Also, by stratifying vitamin D intervention studies based on the individual's vitamin D response index, it becomes possible to enhance the assessment of vitamin D protective role to prevent diseases like cancer and cardiovascular diseases. I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell so you can get any new updates, and thank you.